for this technical glitch at the last moment. And as already Shumon pointed out, it's a great pleasure to have Susmita today, a young and dynamic and very promising researcher who is now Associate Professor at the Indian Institute of Science Education and Research, Kolkata, as an Associate Professor. Susmita obtained her PhD degree from IISER Bangalore after working with Professor Biman Bagchi as her thesis advisor, and she obtained her PhD degree in 2015. And then for 2 years, she did her postdoctoral stint in Lett University, and after that, she moved as a visiting scientist to Los Alamos National Laboratory. And her research integrates theoretical physical chemistry and computational modeling to investigate the sequence and structural dynamics of viral RNA and proteins. Her group has developed a dynamic counter-ion condensation theory for RNA and a structure-based coarse-grained simulation method to explore large-scale conformational changes in proteins, such as the viral spike protein. She has also introduced a Statistical-Mechanics-Guided machine learning method and accompanying web tool for protein mutation prediction. Susmita received the Hargobind Khurana Innovative Young Scientist Award in 2020 from DBT, Government of India, and the CRSI Young Scientist Award 2021, and currently serves as an academic visiting faculty member at the Center for Theoretical Biological Physics, Rice University, Houston, USA. And let me also tell you that so far in SMCB, we had 33 lectures, excluding those special lectures and seminar series. So we are very happy to have Susmita for the 34th SMCB lecture. Susmita, for you now.
Thank you, Ranjit Da. We fondly call you Ranjit Da. Professor Ranjit Beshas from SMCB for kindly introducing me to this platform. So with this, I'm sharing my lecture. Just let me have the moment. I hope you all can see my slide.
Yes, we can see.
All right. So you see this picture that one of the most frontier and exciting interdisciplinary area of research under biophysical chemistry is basically to understand this sequence, structure, function paradigm. You all know that last year's Nobel Prize is exactly what connects the sequence to structure. So you just submit a sequence and AlphaFold 2 will basically develop or construct the structure for you. So in our research group, where we mostly these days focusing on this structure-based modeling and trying to harness structure-based response, try to understand functional dynamics. But today, I will switch the gear towards this way a little bit and try to understand what kind of response is already hidden in this sequence information. So today, it's all about sequence-based analysis. So with that, our journey, research journey on this sequence space actually started since pandemic. And that time we all know that due to the emergence of various SARS-CoV-2 variant that increased global public health risk in the late 2020, World Health Organization classified some of this variant as variant of concern and variant under monitoring, a variant of interest and variant of concern. And this was done to prioritize global monitoring and research and to inform and adjust COVID-19 response to the public domain. So this classification, when it is primarily, can you see my cursor over here? So this is primarily, this classification is based on community transmission and genetic changes. The variant of concern, when it is defined based on the genetic mutations particularly, which is linked to the increased transmissibility, virulence, and often massive epidemiological growth in consequence, which is tabulated sometime in this, tabulated in this table, some of the specific mutation which characterizes, let's say, alpha, delta, omicron, some of the specific mutations, but there might be many which are assessed or known mutations to characterize this variant of concern, which are showing enhanced epidemiological impact compared to the variant of interest. Now, in October 2023, who recommended when close monitoring of this genetic changes is the priority because they are not getting enough current circulating variance epidemiological information from the other side, where they are whether uncertain or some obscurity were there. And then they included a new class, which is variant under monitoring, who actually classified, and this classification, I mean, it's classified other variant, other classifications, but these classifications clearly reflects that these classifications are somehow qualitative, lacking precise quantitative metrics. And this is where our objective of the study underlies, that if we can find some changes when we are traversing from a variant to a variant of concern, and if we can monitor these changes and find an appropriate metric, which I will be defining later as an order parameter, that can reflect the changes and quantify them. So with that objective in mind, first we try to understand the genetic changes, which is also reflected in the target specific protein. Now here our target specific protein is defined as spike protein. Now what is spike protein? Many of you are familiar with the spike protein. That spike protein resides on the viral envelope, and if you split this one spike protein molecule, you will find that this is basically a molecule containing 3 intertwined chains of similar sequence. And if you split one chain out of here, this is called protomer. And there are various complicated domains. I'm not going into the structure-based story today. I will just highlight 2 important domains which can give us certain response, critical response. One is receptor binding domain, which binds to the human S2 receptor, angiotensin converting enzyme 2 receptor, which is on the human cell surface, and another is N-terminal receptor, NTD, which is important or potential antibody binding site. Apart from that, there are various other domains. I'm not going into the detail. Now, we will keep in mind our objective that we would like to understand the changes traversing from a variant to a variant of concern. But before we do that, we need to understand or keep mind of few concerning key points. First is that we would like to understand mutations through which we will follow the changes. But individual mutation event is a stochastic process, meaning that the time and site occurrences are random phenomena that we cannot really underpin. However, their distribution across the genome is not uniform, meaning that there are natural selection is operative, which is not random. So there are positive selection pressure, which is kind of acting as a force that favors certain genetic mutations or certain variant over other in a population. For example, if we go back to that table, you will find that there is this D614G mutations that first emerged in alpha variant and that retains till Omicron it is circulating. Okay, so this is kind of tells about the viral fitness or the mutational fitness, which is living for a longer time. And that kind of information we get often in evolutionary biology in the phylogenetic tree. This has a lot of information. Now, this information we would like to transform what we are familiar with, that how we can, although these are random phenomena, but how we can harness a collective behavior from a random phenomena and the corresponding mutational response we can quantify by any chance when we are traversing from a variant to a variant of concern. Now, when I am saying that we would like to traverse from a variant to a variant of concern, we need to construct, the first objective is to construct a real-time evolutionary trajectory. And for that, this phylogenetic tree is very important, updated phylogenetic tree. This was our first study was in 2022. And we basically, this image was derived from NextVen, which is basically a pathogenic data repository under US NIH. And from there, we obtain this, the real-time information of different family tree. And this contains a lot of information. I'm not going into the detail. This is itself a very specific regime of research under evolutionary biology. But a few features like branches, which basically tells you the evolutionary lineages, the nodes, which are common ancestors, branch length, which is often proportional to the number of mutations. You can get a lot of information from this branch length, how they are going to survive. And the clade, like a group which is having a shared ancestor. For example, for delta, you see that there are a lot of branching, and this is called delta clade. So from here, our specific objective is to obtain the ancestry level information, means the ancestor and the following VOC when that emerges, and that time series data, time trajectory data. So exactly, we did that. First, under this objective, what we did, we constructed multiple sequence alignment, and this MSA helps us to assess the randomness. And as a statistical mechanics practitioner, what we could think about, the first quantity to really quantify the randomness is nothing but entropy, right? So we basically refers to it as a mutational entropy, but we used a very simple Shannon formula, communal entropy formula. And here, basically PJ stands for the likelihood of a particular amino acid in a protein sequence or nucleotide for a genomic sequence that will occur at a specific site. And SI is the Shannon entropy for site I. So this is site-specific entropy. The range of the potential amino acid or nucleotide is represented by this index J, right? And J is the member of proteins group, and you know this 20 amino acid and genomic sequence. So this is not only helps us to understand the site-wise mutation, but the site-wise mutation could be a mutation hotspot detector, because you can understand those domain-specific patches where mutation is more probable, meaning that natural selection is operative, and you will see a lot of heat, but maybe all heats are not equivalent or equally important. Maybe among all those heats, one of them turns out to be functionally important, or two of them you can identify after a mutagenesis. So this is basically important before we conduct even mutagenesis experiment to identify what are the mutational hotspot in a very complex protein. So now, once we find this, what we did, we basically plotted this entropy along the real time. And this is basically, you can follow that this is a B1 ancestor for delta. This is for a case specific, I'm showing you that first delta, we are following from November. This is a very limited data I'm showing, but we have actually constructed a very long time information in the ancestry level. So here you see that delta basically emerged in February 2021. And this information, particular information, we collected from many various public domain like GISAD, NCBI, all this kind of data server, publicly accessible data server. So here you notice a lot of changes. If you just analyze this, you will find that the entropy fluctuation basically in the month of November to January retains over in this region, which is at the intersection of RBD and DD. And then once delta emerged, it is shifting to the NTD domain. And I'm reminding you, this NTD is basically this antibody targeting binding site. So this is basically tells us the human host cells immune response, that it is delta is really affected us. So now, once you get this data, next what we ask that how to really quantify this transition, meaning that this transition, which is a non-equilibrium transition, because you see that this is a time-dependent phenomena. right? So how to quantify and then rationalize this time dynamic transition of mutational response. So whenever we see this keyword, the statistical mechanics practitioner immediately redirect you to this particular chapter in the textbook, which is fluctuation and response function, phase transition. And particularly from where I have learned, I would like to suggest you the following chapters are now in a form of chapter in this particular book by Professor Biman Bagchi. Chapter number 6 is fluctuation and response function, which helps you to relate that how fluctuation is connected to various response function in a form under the external perturbation. For example, energy fluctuation relates to the specific heat, volume fluctuation to the transmissibility, magnetic susceptibility, magnetic fluctuation is to magnetic susceptibility, and so on and so forth. This response function often undergo dramatic changes or tend to diverge near the critical as you near the critical point. So you are familiar with this phase transition graph and as well as this PV diagram, which is in Atkins. So this is particularly, these slides are for students and how they are going to now understand this response function. Now this response function and their dramatic behavior in this coexistence line, for example, this gas-liquid coexistence line, or as you approach the critical point, is particularly help you to understand, or first, what you would like to understand, the that what property will be help you to harness this response function. And this response function, when at the critical point, basically this motivates you to understand the order from the order parameter aspect. It motivates you to identify a suitable order parameter which is sensitive to the changes and capture some early warning signal. For instance, in gas liquid transition, you know that the density the difference between liquid and gas, how it is a natural choice to understand the phase transition between undergoing from gas to liquid. Now, as you are, suppose that you would like to understand the phase transition from paramagnetic ferromagnetic, the net magnetization is the natural choice for understanding phase transition. Now, phase transition thermodynamically often described by the discontinuity of the derivative of the free energy, which justify the concept of latent heat. Now, this is the first attempt to classify phase transition is by Professor Ehrenfest, and he actually classified this as based on the lowest derivative of the free energy. When the first derivative of free energy shows discontinuity, we refer to it as a first order phase transition. Example, solid liquid, liquid gas. Here there are lots of features that latent heat is involved. You will also find hysteresis and metastability if you critically analyze them. In the second order phase transition, however, first order derivative is continuous, but the second derivative shows discontinuity. For example, ferromagnetic transition, lambda transition in helium, but here in the second order case, there is no latent heat involved. In this classification, you can also find higher order transition, but this is kind of a hypothetical and rare in real systems. So this classification particularly shows a limitation when we are trying to understand critical phenomena near the critical point, right? So the modern approach and the central theory for the modern approach of classification underlies in the Landau theory of phase transition under the equilibrium statistical mechanics. And the basis is that you understand the nature of order parameter, symmetry and divergence of the order parameter. And based on the order parameter, you try to attempt to classify first order phase transition and second order phase transition. Landau theory is basically a very simple Taylor series expansion as a function of order parameter. So this is a free energy functional. You try to understand the symmetry of your system of concern, and then you expand this. And the following recipe is quite formal, that you minimize the free energy. then you will get to the solution of below critical point and above critical point. And once you plot the order parameter variation as a function of temperature, you will find that in the case of first order phase transition, at the critical point, order parameter shows discontinuity. And when in the second order phase transition, you will find that order parameter varies continuously. So if you derive this very formally, you will find that the variation of the order parameter as a function of temperature. Now you also know that the second order derivative of the free energy gives you response function. So this is fundamental thermodynamics, and that gives you specific heat. And once you plot the specific heat as a function of temperature, you will see in the second order will show a discontinuity as you approach the critical point. Okay, and sometime it diverges if your system is infinite. So now, this Landau theory, if we summarize, you will find that in the equilibrium phase transition, when we analyze fluctuation and response, this response we get as a function of temperature. And we came across then a very recent paper that instead of temperature, you can also vary time and then can understand under a non-equilibrium situation a dynamical phase transition. And Professor Esposito in this paper, and this paper also came in 2022, that they understood the dynamical Landau theory of phase transition, and they derived this. So this is the paper that little bit I'm going to briefly discuss, because this gives us the rationale and what actually motivate us to understand the collective mutational response in this context. So what they basically did, that they took a nearest neighbor Ising model on a 2D square lattice with the PBC, where we can basically write the potential energy function. As you are familiar with this spin-spin correlation, this is Ising model, very classical Ising model. So this is spin-spin correlation, and what they define that as an inverse coupling temperature, where J is the coupling constant. And magnetization, they are basically taking magnetization, accounting the finite system size. So this is the dimensionality, and this is the length of your system as a summation over all spin, and equilibrium magnetization you are getting after ensemble average. So now, under the this model shows a continuous phase transition, and the vicinity of the critical point, the several thermodynamic quantities diverges, and similarly you can identify that the fluctuation, which is your susceptibility, also diverges. And just give me a second, because this photo is, zoom picture is hiding my screen a little bit. So the order parameter shows a scaling behavior where beta is the scaling exponent, and this is 1/8, and you get this exact solution from Unsager's explanation of this solving this NNIM model of this 2D Ising model. And close to the critical point under the equilibrium features, it also diverges where gamma is the critical exponent, and this Fischer has an exact solution where he has calculated that the gamma should be 7/4, which is associated with the critical exponent. Now, in contrast, in the dynamical phase transition, what Professor Esposito designed is a very unique non-equilibrium situation. What they did, they quenched a large but finite NNIM model from an ordered to disordered phase, and they monitored time-dependent rate function. Now, this time-dependent rate function they obtained from large deviation theory of statistical mechanics. What is this large deviation theory? This large deviation theory is nothing but the generalization of the central limit theorem. If you see the Gaussian distribution, this rate function on the equilibrium situation is just equilibrium fluctuation, the exponent in the Gaussian, but now it is a time-dependent rate function. So this is basically A by Cramer, first time derived by Cramer at the when the system size is infinite. So in the non-equilibrium case, now for finite system size, what Professor Esposito is deriving, I'm not going into the detail of the derivation, however, that here time plays the role of the inverse coupling temperature. And they also have derived that similar to the equilibrium phase transition, the susceptibility diverges near the, now not near the critical temperature, but near the critical time. What happens that here, the initial rate function, which is analogous to your free energy function, now becomes as a time-dependent function. Initially has a double well-shaped potential with two consecutive minima at one m, some m plus q and minus m eq equilibrium. And as time evolves, the minima approaches to the origin and a sharp king forms. Cursor is not working sometime, but this is not form. You can see that, right? My cursor? That there is a king form at m equals to 0. And this implies that this king is associated with the large deviation of magnetization generated by rare relaxation event where system relaxes to the vanishing magnetization at m equals to 0 in shorter time. Now you do an experiment that you shoot your trajectory, let's say, and calculate the probability density. You shoot your trajectory from some m average and then calculate the probability density that how many are reaching to m equals to 0. At a shorter time, you will find that the probability density is 0, but beyond a critical time, this probability density will reach to a finite value, which will reach from some m average to m equals to 0. Now, this critical time behavior is quite analogous that I have just shown you for the mean field model of Landau theory. But nonetheless, here also we find that the response function around the critical point, same like equilibrium phase transition, shows discontinuity at the onset of dynamical phase transition. Here there are more to it because they will be now showing critical scaling behavior, and this is for what they are analyzing is varying the system size, and finally with the scaling they will show that all this curve will converge, but the behavior is quite similar. But this gives us the rationale that yes, we can write a free energy functional as a function of time, and as we have seen in equilibrium phase transition, that how response function behaves near the critical temperature. Now it has an equivalent natural variable, which is now time, a critical time. So this motivates us to introduce a mutational response function, which aptly quantifies the mutational entropy fluctuation, and which is basically when we plot it along the real time, this mutational response function shows discontinuity as we transit from beta B1, which is the ancestor, to the delta, which is analogous to the dynamical phase transition, reminiscent of Landau theory of dynamical phase transition. So you see that this is basically delta. And here we particularly focused on that entity region which has a better response domain, or this is basically gives us the immune response in terms of antibody binding. So you see that this is the result for delta, and this is for Omicron. And now this is, we can understand from physical chemistry concept, but as well as this is biologically also relevant if we just see this timeline information, that delta was first detected in India in February 2021. But the genomic level response became evident in January 2021. And who declared delta and its sublineages as a variant of concern on May 10, 2021. So this is what basically, this is where we badly need a quantitative metric that could serve as a early warning metric for the emergence of a variant under monitoring, or maybe variant under concern, if we can refer to. So after we understand the collective behavior, and when we understand that entropy basically serves as a very good filtration metric to identify the mutational hotspot, or even in a complex protein, we try to understand that can we really predict, if AlphaFold 2 can predict sequence to structure, we thought of, can we really predict sequence, learning sequence to a new sequence, or a new mutation rather, by very carefully learning the mutational response. So with that, basically, we started this supervised, developing this supervised machine learning model back in 2022. Actually, this is that time ongoing work. And we trained this ancestor level entropy data. But there are other features I'll be just talking about to predict mutational hotspot of a progeny. So this is basically the origin, how basically we develop, what is the motivation behind the development of evolve, and it's an acronym in real sense, that this is evolutionary variant observation learning and variant of exploration. And there are lots of features, technical features. I'm not going into the detail, but certainly the challenges that we overcome computationally is the key capability of handling the vast protein sequences, because you need to pre-process the data a priori before you understand any metric, quantitative metric, and basically design a new mutational landscape. So the methodology is like this, that you first make the data, collect the data, and the data it will be basically goes to a data processing module. And first, what it will do, it will clean the raw FASTA input. These are very technical things, but I'm just reading out that it will perform multiple sequence alignment. Then it will extract the features, important features. We have some delineated features. So here I'm going to talk about the features, but then it will integrate the machine learning. And then you can basically, we have a very user-friendly tutorial with which you can also analyze a lot of the properties, and we have such analysis module. Okay. So the feature one is basically pair predictability matrix. So this is motivated, this motivation we got from AlphaFold 2. You know that AlphaFold 2's main features, which is dictating the structure, is coevolutionary information. This is some sort of similar coevolutionary information matrix, but it is pair predictability. It is a very simple joint probability, because we thought that it is important, it will be important if a single point mutations affecting an amino acid at a specific position, this can also lead an alteration to the amino acid pair, consequently resulting to a distinct pair frequency. So this is a pair frequency. This is our feature number one. Consequently, we have 4 features. The second feature is entirely inspired from statistical mechanics. This is kind of how, this is essentially Boltzmann counting, the similar kind of microstate probability of the particle, which is occupying energy state following equal a priori probability, and then we calculate the probability of those states, each state. So here also, as amino acid within a protein exhibit a non-uniform distribution characterized by a kind of cluster of a specific region, then homogeneously spread along a sequence, then that spatial clustering tendencies of the amino acid across sequence reflecting local neighboring distribution, which may shift due to the mutation. And those distribution probability we can calculate by this multinomial formula. So this particular term is basically degeneracy of the macrostate. This term is the multiplicity of the macrostate, and this is where we follow equal a priori probability. So here R N is the number of partition, R is the amino acid at ith partition, all right, and QI is the degenerate partition with the same number of amino acids. So this is basically degeneracy or grouping. And N to the power minus Ri is when your probability following equilibrium is 1 over N, R is the total number of amino acids. So then third feature is the future composition. And this is also important because we have a codon table where we have an expected amino acid which are being synthesis from the 3-letter codon. And you know that we have 64 codons. So this feature particularly focusing on the examining the amino acid composition within a protein sequence and how it changes due to genetic mutation. And this analysis is geared towards estimating likelihood of a specific amino acid mutating into another amino acid, a calculation facilitated by the insight of the RNA codon table. And thus, this mutational probability is kind of a ratio that the predicted contribution by actual contribution. So with this, the fourth feature I have already talked about is entropy. And with this fourth feature, what we did, we basically integrated a multi-layer perceptron model. And here, this is basically a very well-known algorithm, deep learning algorithm. And if you would like to learn in detail, you can find in this link. But how we actually developed the algorithm following these steps, that we first computed a linear combination, which is weights and biases to your features. And then the data went through an activation function. We used this tang function. And then we calculated the loss function following this binary cross entropy loss function. And then we had a back propagation. We computed the gradient of loss. This we have not explicitly shown, but this is kind of an iterative process. And then we optimize the weight using the stochastic gradient method. So this is a very brief MLP. This is basically the you know that this is input layer, this is hidden layer, and this is output. And we used a very simple, very minimalist approach, which is 4881 feed-forward neural network architecture. And then is the turn to get the result, which is these days how this machine learning works very magically, to get very accurate results. So this is basically where the ML model is able to predict the future mutations, which were not in our training data set. The ratio between our training and testing data set is 85-15 kind of a ratio. And here for delta 2 crucial mutation, we have predicted this around 141 and 681 among others, which are directly reported to enhance fusogenicity, meaning that it is basically improving the entry mechanism of the spike protein of delta variant. This is Omicron result, and the precision level right now. We are calculating precision because here we think that the false negative is kind of obsolete, and then the model predicted out of 13 positions, 9 positions marked as confirmed mutation by NextTrain. So again, NextTrain I have already talked about, that this has database of the viral genome with their pathogenicity. And the new mutation, particularly this green color, this one particularly, this particular green color, this is not appearing in a correct color A, but appeared in December 2024, which is confirmed by NextTrain, but this position was already predicted in our 2022 paper. It was a JPCB paper. And importantly, we would like to highlight that this model, however, was trained exhaustively on the sequence of original Omicron variant, withholding all the data on Omicron subvariant. When predicted on this subvariant, the model identified 13 potential mutation sites. And 4 predicted mutation sites, which are highlighted here, I'm not reading it, had already acquired mutation in the circulating Omicron variant. So this is, we are basically validating it, that accurately it is pinpointing those mutations which were not in our testing data set, but it really appeared during the natural evolution. First of all, there are two aspects. One is that some mutations for which next strain has the data about their functionality, about their pathogenicity, but some mutations which are found in the natural evolution itself. So which is important, that which gives us a lot of confidence that these 6 predicted mutations are annotated in the next strain as well. So then we basically went beyond analyzing SARS-CoV-2. We have analyzed with Evolve, we have analyzed influenza, Xanthomonas oryzae, citrus citreza virus. This is a plant virus. So with their target proteins, and we found very good accuracy in terms of precision. Sorry about that. Particularly, what is important in explainable AI, that you understand the importance of the feature and you would like to take care that it is not a kind of overfitted result. So here we particularly analyze that by adopting a lasso regression analysis. So it is a linear regression analysis. Again, the detail you will find in this link, and particularly we found that when we are training the data, entropy is really behaving remarkably, leading the data. But then we did a lot of analysis to understand these features, because we have only 4 features. So this is very explainable and understandable. So we tried to understand, and then we found that this feature 2, which is basically the distribution probability, is not much contributing. So when we left that, then we found that all the other is normalized accordingly. And now pair predictability is also contributing quite efficiently. We have a very unique module, and particularly this module in Evolve, we have designed when we faced the scarcity in data. This is a very unique methodology that we have, or strategy we have developed, that particularly we faced for dengue and Ebola, where data scarcity is there. What we did through MSA, we tried to find that if there is 85% cosine similarity, I'm giving you the equation. If we find the cosine similarity in terms of the probability of the similarity index, then we included that in our training data set. And once we do that, then we found that for dengue NS3 protein, the precision level earlier was quite bad, but now precision level is 70%, quite acceptable. So this is kind of a table for animal viruses, that what is the precision level. But for Ebola, you see that if data is poor, then the precision is, is not kind of acceptable region. So now another thing we thought about, that we are predicting new mutations, but suppose that we are predicting a very novel new mutations whose data or whose pathogenicity is not at all known. So what we are going to do that, how we are going to really defend that, yes, we are getting a relevant mutation, functional mutations. For that, little bit classification we need to know, a very simple classification of mutations. So there are various kinds of mutations, but mutations are majorly classified as functional. This is a terminology. Functional mutations are missense of type, then benign or neutral mutations, and then other extreme is nonsense mutation, when there is a loss of function. So recently we came across, this is a 2023 paper by the same group, by John Jumper and Hasabi. They came up with this alpha missense wave tool, a new methodology where basically they are taking this input of a sequence, and they can predict its pathogenicity. So what they are pinpointing is a very important message, that of more 4 million observed missense variant, meaning that functional variant, because there are many mutational data, only estimated 2% has been clinically classified as pathogenic or benign. means they are assessed, while vast major of them are of unknown clinical significance. And this limits the diagnosis of rare diseases as well as development and application of clinical treatment that target the underlying genetic cause. So this is really alarming that we do not know at all that what mutations may cause a rare disease or how it is related to the pathogenicity. So this, we are also linking this alpha missense, integrating it in our pipeline now. So summarizing Evolve today, that we have a very good tutorial, and that will help you. If we could show a demo, that will be wonderful some point, but this is the science behind Evolve today, that if you submit the bunch of MSA, it will pre-process the data, it will calculate the feature for you, then it will predict based on your data set. Of course, the data should be quality data. And then we are confirming the mutation, predicted mutations, potential predicted mutation, which are novel by Nexttrain, Fluserver, EV coupling. These are all available data server. Available tool actually, which will help you to understand the pathogenicity. You also check the stability, and the stability analysis you perform using this bioinformatics tool. And last but not the least, if it turns out to be a very novel mutation which are not reported in Nexttrain or in this server, then you can predict its pathogenicity using alpha missense. So this whole pipeline you can integrate with Evolve and analyze. So summarizing everything, finally, I would like to thank my students, and particularly the lead author of this Evolve, Mr. Satyam Sangeet, who has now moved from our lab to Australia. He's in University of Sydney. And Anushree is the second author. She also helped a lot in this process. So Mr. Raju, Sarkar, SRF, Madhav, our MS thesis student, he actually, his work was also ongoing, that we are also looking into designing this covariance, meaning that the co-evolutionary information, and if we can design, because we are now designing a single point mutation, if we can design that, then we can directly connect to the structure, or at least try to understand the structure-based implication. And Orpita, she was a summer student, but she also helped along the process. And finally, I would like to acknowledge my funding resources, DBT, Hargobind Khurana Young Scientist Award, DST, SERB, Startup Research Grant. This is an Early Career Research Award, and currently ongoing National Network Project in collaboration with University of Pollani and IISER Kolkata, certainly for infrastructure. So thank you for your kind attention. I would be happy to take any question if you have.
Thank you, Susmita, for really fascinating talk and fascinating work. So this establishing the connection between statistical mechanics and the sequence-based data is really novel and very cool. So I'm sure there will be some questions, and so I request all the participants that you can use the React button below to raise your hand. So if you raise your hand, I will ask you to just come online and ask the questions directly. Meanwhile, there are a couple of questions in the chat box, I think from some students. So there are a few pedagogical questions. So I'll just read those questions for you. And yeah, and so the first question is from Abhinandan. What is the difference between mutational probability, mutational entropy, and mutational biodiversity?
Oh, very fine difference, probably. Mutational probability, you see that, let me show you the equation that will clarify and answer your question. Yeah. So you see that this P is nothing but for a specific site is your mutational probability.
Okay, let me also mention the second question because that involves this Pij. So Shahira asked, in the Shannon entropy formula, what does Pij indicate? So maybe you can clarify this point and go further.
Yeah. So for both of your questions, this will address your question, pretty sure. That you see that this is the entropy for ith site. But Pj, what is J? J is basically summing over. This is the index for each amino acid. Or if we are calculating for in the genomic sequence, this is for each nucleotide. So taking J is nucleotide, which is the sum over, and I is the index, which is for entropy. So I is not included in my summation iteration. Does that answer your question? Okay, I think this answers your question. If not, you can come up. Okay.
Yeah, those who ask the question, feel free to just directly
interact.
respond. Yeah, directly interact. Or just put it in the chat box if you have any further questions.
Okay, so another question, I think the first question
differences between mutational probability,
mutational probability, and you see the mutational probability, this is entropy. And another thing you asked, that biodiversity.
Yeah.
So biodiversity is basically heterogeneity, but mutational biodiversity is kind of a sequence similarity. And this is basically what you calculate from taking the look at the entire MSA. Right? Taking the look at entire MSA. So you calculate the sequence similarity to understand biodiversity. But this is a site-wise entropy, site-wise probability. Okay, so this is what the fine difference between.
Yeah, I hope that answers the questions. If not, feel free to directly interact or put it in the chat box. If anybody else has any question, please feel free to come forward. Any comments?
Yeah, can I ask?
Yes, Biman, please.
Yeah, so I'm just thinking that right now you are probably, if I understand correctly, you are probably predicting the which position will be more probable to mutation.
Right.
Not really telling that what will be the mutation or what kind of mutation it is going to give you.
That we are predicting with some uncertainty, though. We are predicting from the codon table.
Predicting from what?
From the codon table.
Codon table. Okay, okay, yeah, I got it. So that gives you what amino acids it will be
switching to.
Okay, okay, yeah, that's what I wanted to know.
Yeah, but mostly Evolve predicts what site is the most probable future mutation.
Mm-hmm. And also, just one more addition to this is that it is okay to predict a mutation, but then in nature, whether a particular mutation or few mutation together will survive as a strain, that is a very difficult question, right? That means I'm asking that I have a sequence and I predict a position and predict a mutation, let's say, but that single mutation together might not be enough for the survival of that strain. So then that will not be presented, right?
Yeah.
So I
mean, there are lots of already existing understanding. Somebody is really writing on my screen. Okay. Zoom is weird. So anyway, going back to your question, Biman, that there are quite a few existing well-known understanding in evolutionary biology that the mutational, how you are going to quantify mutational fitness. So so there are quantitative metric which is identifying synonymous and non-synonymous mutation. Synonymous mutations which are going to, first of all, synonymous mutations are which are reflected in the as a genetic mutation, but then it will be converting when during protein translation as a protein mutation. Okay. Then as a protein mutation, when they whether they will be surviving or not, there is another terminology is called advantageous mutation. Maybe it is connected to the function, which is missense of type, but whether that will be advantageous or not for the fitness, overall fitness of that strain, spreading in the population, that is called advantageous mutation. Similarly, in the non-synonymous mutation in evolutionary biology, non-synonymous mutation means you have the genetic mutation, but that is not translated into a protein mutation. And then suppose that is a non-synonymous. In the synonymous also, if that come in, that may delete in the protein sequence. That is called deleterious mutation. So there are this kind of terminology one can assess from the MSA.
Okay.
And from phylogeny also you can understand the length, the ages are basically gives you the survival, the length of survival of a variant, particular variant.
Yeah. I had a kind of a general curiosity. Actually, it's part of the discussion that we were having. So right now you are just using the sequence information to predict, let's say, the mutation and all. So this entropy, it essentially captures the sequence information of your this protein. But when it comes to the fitness, that actual biological function, so that would involve the partner as well, right? So where it is interacting to or where it is binding. So is that information, can it be important for functional fitness in some sense? So here we are looking at just one sequence evolution, right? So I'm just curious that how much of this entropy is capturing the functional information.
Right.
Yeah. Or there could be some another parameter that could be connected to the actual biological function.
Exactly.
Can I say something?
Yes, sir, please.
Okay, I think one problem I can see in this definition, which somebody already asked, I believe that this will require more detailed entropy, such as it is a site entropy, right?
Yes, sir.
You might need two particle entropy, that means correlation. Like we do radial distribution function. So we have an entropy at the level of ln Shannon entropy, ln P, ln P. Now there is a higher order entropy, which was done by Green's expansion, which actually should be done in this kind of systems. That is H.S. Green, not M.S. Green. 1950s, Charushita used extensively. That is, entropy is integration dr gr ln gr. So that's an entropy where you are higher order, where correlations are there. I believe that you would need could be a zeroth order or first order. Higher order you would need to consider. I always thought codon is not enough.
Yes, sir.
What does it have surrounding it? These people talk a lot about the junk DNA part, which seems to be having importance. So higher order correlations could be important in mutation. For example, if you look into lung cancer or all those things, then there are some very specific mutation, which is some deletion of something, exon 19. Now, those mutations are exceedingly important because they almost, now this is possible in the last 10 years to do the second molecular test of, and where you get all these, cancer is the best place for you to work actually. All these mutations are available and these mutations have function because these mutations are then immediately connected with those proteins which apoptosis doesn't happen. Apoptosis does not happen because of malfunctioning of the proteins. And the proteins malfunction because of this mutation in DNA. Okay. So now when I'm going to this protein, codon protein, this DNA protein, the transcription business, there one has to look into the relation between them, and I don't think the relation is single site relation. It would probably be multi-site relation. That means why I am having, if I am a smoker, why I am having this. This is the famous paper from Weizmann Institute. I don't know whether I got a Nobel, but it should have got a Nobel, which they showed that smokers have this kind of mutation. Okay. Now, I also smoke, and hopefully I don't have many mutations. Now, many people smoke, and only 5% of them get the mutation, or 1% get the mutation and goes over to have cancer. So, mutation depends on so many things. It is not a single site property. It's several properties, several higher order correlations. I don't know how to build those correlations.
Sir, what I am currently trying to adopt is a Potts model, and then trying to calculate the higher order using covariance, actually, a matrix which is
covariance is the same as the ideal distribution function.
So I am trying to use that, but that is today I could not really cover because it is completely not. It is an ongoing.
Sure, but I think that would be the way to go. That biology would certainly require many site correlations.
Yes. For now, we have a prior predictability. This matrix, it's a joint probability, basically.
I think there's a lots and lots of data becoming available, at least in one cancer, that is two cancers, and breast cancer and lung cancer. So I think you would need, I don't know this, you have AIMS next to you, but those AIMS, what I hear, is not a great place. But if one can get you data, many data are probably already available.
Yes, sir. Yes, sir.
This is only, this kind of molecular testing. I was reading a webinar a few days ago by one of those oncogen guy. He was talking that this whole thing has changed it from early this century, 2005, 2002, around that time it started changing. Because they have now been able to find the mutation, in DNA. And now they know that because of this mutation in DNA, this protein, I have to target this trypsin. They call, I think, KTI or TKI, that particular A. That, I think, is one of the best area of research. It's even if you don't be able to come up with anything great, it's worth working, because that, and I don't think anybody in India working too much, except the CAR T-cell therapy is doing great. And another thing, you should get in touch with those guys in Bombay.
Yes, sir. Some of them are connecting now after this event, because they would like to understand this mutation business in oncology as well.
By and large, I think you should probably quit everything else and work on these problems, because these are the problems which most of us do useless work, right? I write in my blogs. But if you get a chance of doing this good work, then you should pursue it. And yeah, okay. This is a good way to go.
Thank you, sir. So before I just continue with my question, there is another comment in the chat box. I think it's a kind of a technical question that, Paramananda, if you are here, you can ask it directly. Otherwise, I'll just read. How can we achieve a real-time performance in evolutionary algorithms with high computational demands in current and future times? This is more like a computer science kind of question, probably.
So that they cannot have. I am working on origin of life now. This real time, forget. Then you have to wait billion years.
Evolution, yes. To answer your question, let me tell you that evolution is really a slow time scale process. Really, really slow. So to get the real time performance, at this time point, what I can just redirect you to one of my plot, if that can answer your question, but not concretely. Just a minute. but let me try. You see, after this dynamical phase transition, when we came across this plot, and I made a comment here that the timeline when we are predicting the genomic data is much earlier, means genomic data is showing the response is much earlier than who. So at least what I can tell you that how accurate we do not know, but data, this is real patient level data, right? At least reflecting, and this is a dynamical, these are exact solution. So this is showing that we are predicting pretty earlier than who is declaring. So the accuracy, computational accuracy, I cannot really comment on. By any chance, my student Satyam, I can redirect you to him to understand that if he can tell you more about the technical part. But at least biological relevance wise, this timeline is important. And we are much at the sharper regime when we are applying statistical mechanics. If that somehow, somewhat partially answers your question.
Okay. I think I'll finish with
Am I audible?
Yeah. Yes, you are audible.
Yes, ma'am. Hello.
Yes, Paramanando.
Yes, ma'am. I just want to ask that can I get the paper you have just showed? Can I get what is the source of this paper? Because
Yes. Let me
chat box, if you can paste the link
in the chat box. Certainly.
Okay, ma'am. Thank you.
Thank you, Paramanando.
You can always email Susmita, and I'm sure she'll be happy to answer. You can email her. Yeah.
Okay, ma'am. Okay, sir. Thank you, sir.
Yeah, so quick. So Susmita, so maybe I'll just end with my question out of my own curiosity. So it's a kind of a naive question. So when you are trying to predict new mutations, like finding the arrow of time and also the strategy of the virus where it should go next. Now there could be the strategy would be related to the functional strategy, right? So for example, some viruses like let's say Ebola, this has high mortality. So it will kill quickly, but then it cannot propagate further. Or like COVID, for example, has low mortality and it can high infectivity. So I'm just trying to think that there can be different strategies how the virus will survive. Now the mutation can take different approaches. Now your entropy, whatever it is trying to capture, this arrow of time. So which I'm just trying to understand that there could be multiple arrows of time or multiple pathways or strategies, right? To optimize its strategy of survival. Now, you are getting probably only one directional arrow when you are trying to predict. But how is it connected to this many possible strategies that how the virus might survive? Am I clear? I mean, I'm just trying to think that how your arrow of time, how is it connected to different possible strategies for the virus to survive functionally?
Very when you are adding this last word functionally, which
because when you say this kind of classifications like missense, nonsense, etc., that those are also functional, right? So
certainly. So what we are, let's say, first approximation that I would like to take upon is that the mutational probability, the hit where we are getting is a very clustered patch on a particular domain, which means what? That if we try to find out or build a mutational landscape where you can see that there are several possible pathways along the arrow of time. So this is basically a pathway connecting, right? Sort of. So we are now currently analyzing. In fact, Shumanda, I wish I could have.
Essentially, I was thinking like a pathway. If it's a landscape, there should be multiple pathway of. It is not just one pathway, right?
And I have shown you the dynamical phase transition, the similar approach, that how in a time dependent way now free energy changes. So if we can have a local equilibrium somewhere for a variant, it is somehow during its survival. If we have one kind of free energy, then in a different time, we have to cluster in accordingly, that in a different time, we can get a different kind of free energy landscape. So this is how we can check the local. And in fact, we are showing that now, that suppose that some mutations or some region which are for some time is conserved, which sometimes we need that the conserved region for drug targeting. But this conservedness are disappearing if you follow them along the timeline. So this we are getting as a result now, it is an ongoing analysis, that conservedness is also kind of a very limited time phenomenon. If you move along the timeline, if you have enough data, what we are actually analyzing now, and we are getting, Satyam is having the data, that one particular region which was showing conservedness in the timeline, that conservedness is now turning into a dynamical region. It's now mutated. So it's a very time-dependent phenomena. One can build a time-dependent free energy, like Professor Esposito constructed.
Ma'am, I have another question. Can I ask?
Yeah, maybe quickly.
Yes,
Quickly you can ask. We are running short of time. Yeah, please.
Basically,
Yeah.
that's a wonderful session. So I have a data related some questions, like how we can literally handle the data scarcity or the quality issues in the real-time evolutionary modeling.
The answer is follow our. I have shared the paper. I do not know whether you can see this link where you will find transfer learning module. So follow the transfer learning module, how we have adopted cosine similarity, and there the data scarcity can be handled.
Yeah.
Follow the pipeline, okay, on the transfer learning module.
Okay. And there is one last question in the chat box, but it's a more COVID-related question. So why COVID-19 was on peak in November, December, and June always? Is there any particular effect on temperature or some other environmental conditions?
Environment, weather, host response, so many things. This is kind of a question that how boils down to the question that which environmental factor is associated with which kind of mutation. That is also an interesting regime to explore, actually, with this ML. You can connect. So there are a lot of unexplored region one can explore in this. One can connect environmental factor to this. Well, that could be another feature, certainly. It's a very interesting question.
Yes. So thank you, Susmita. I think so. There are some more questions coming, but
popping up a lot of questions now.
So we'll have to stop at this point. It has been.
I will take this question. I will email Omkar.
Yeah. So for all further questions, I request you to please contact Susmita by email. I'm sure she will be happy to resolve all your questions. So yeah. So maybe you can, Susmita, if you want, you can.
Shumanda, just a request, because I think the students did not receive the paper. Let me just share the link. They have mailed in the chat.
Mohanty is asking for the paper link. So if
so
want,
let
you
me
can share
right
the
away.
link. Yeah, I think Satyam has shared already. Yeah. Okay. So now I think Paramananda, you have found the link, and feel free to contact Susmita in future for any further clarifications. And also for those who could not attend today's lecture, it is already recorded and it will be put on our YouTube channel. Many of our earlier lectures are already available on our YouTube channel, and feel free to explore our YouTube channel. We have a mailing list which is mentioned in our website. So please feel free to register in our mailing list to be to receive all the future announcements. So once again, I thank our today's speaker, Dr. Susmita Roy, for a very fantastic scientific discussion. And I thank all of you for staying here till the end, and looking forward to meet you again in the next SMCB seminar. Thank you all. Thank you, Susmita.
Thank you.
Okay. Bye. Bye, everyone.